Hello, bachcho. Let's see the question. So the question says manipulation of DNA in genetic engineering became possible due to the discovery of. So इन चारों ऑप्शन में से हमें वो आइडेंटिफाई करना है जो मैनिपुलेशन ऑफ डीएनए में सबसे ज्यादा मदद करता है. So the key concept in this question is to understand genetic engineering. So genetic engineering क्या है? Genetic engineering is actually the alteration or the modification of the gene of an organism. So ये एक ऐसा टेक्निक है जिसके मदद से we can modify the gene of an organism. So genetic engineering evolved with the discovery of a very important technology which is known as the recombinant DNA technology. So in this technology, we can add the gene of interest into any host organism in which we want. And therefore, with the help of this technology, we can create something called recombinants. So recombinant DNA is that DNA which involves the DNA of two different species. So एक recombinant DNA में दो different species के DNA present होते हैं, तो उसको हम लोग recombinant DNA बोलते हैं. Now, the discovery of this technology was only possible due to the discovery of a very important enzyme which is known as the restriction endonucleases. So restriction endonuclease enzyme सबसे पहले बैक्टीरिया में डिस्कवर किया गया था, and in bacteria they play a role of cleaving the viral DNA, and therefore they are used to fight against bacteriophages in case of bacteria. So the main function of restriction endonuclease is to cleave DNA at very specific sites which are known as the recognition sites. So restriction endonuclease can recognize a particular sequence of DNA and cleave the DNA into two fragments by acting on a very specific site of the nucleic acid. So this is the function of restriction endonuclease. Now the question arises, how did the discovery of restriction endonuclease help in the recombinant DNA technology? So if we see the basic steps of a recombinant DNA technology, then by using a very basic example, then we can understand the steps as follows. So consider this as the human cell and this as the DNA present in the human cell. And this we will consider as the bacterial cell, and this as the plasmid of the bacterial cell. Now our first step will be to isolate the DNA from the human cells and the plasmid from the bacterial cells. So the DNA from the human cells will be isolated from the cell, and the plasmid of the bacterial cell will also be isolated from the bacterial cell. Now the next step will be to cleave this DNA with a restriction endonuclease. So the restriction endonuclease that we will use will result in cleaving the human cell DNA, and we will get a fragment of DNA which will depend upon the type of the restriction enzyme used. So this will be a very specific sequence of DNA. And then we will use the same restriction endonuclease on the plasmid as well, and therefore we will get a sequence of plasmid which will be removed from this plasmid. So that DNA will be removed from the plasmid and we will have a region which is empty in the plasmid. Now this sequence can join with this sequence if we add an enzyme which is known as DNA ligase. So in that case, when we add a DNA ligase to this, the plasmid and the human cell DNA will fuse and we will get a recombinant DNA. So here we can see that the two DNA, that is the human cell DNA and the plasmid DNA have fused, and so this is known as the recombinant. So the recombinant is formed because because we use the same restriction enzyme for cleaving both the human cell DNA as well as the plasmid, which resulted in the complementary sequence end at both the DNA, and therefore when we use DNA ligase, the two DNA fragments joined together and it resulted in the formation of a recombinant. So in this way, we added the gene of our interest into the bacterial cell plasmid. So this is the very basic step of recombinant DNA technology. So the main enzyme which plays the most important role in recombinant DNA technology is known as the restriction endonuclease. So if we come back to the question, then the first option says restriction endonuclease. So this is the correct option because with the discovery of restriction endonuclease, it became possible for the discovery of genetic engineering. The second enzyme is DNA ligase. So DNA ligase is also an important enzyme for genetic engineering, but its discovery did not lead to the discovery of genetic engineering. So this won't be the answer. Third option is transcriptase. So transcriptase is not involved in recombinant DNA technology. So again, this is not important. And primase is used for formation of primer, which is also not required here in recombinant DNA technology. So our final answer, so हमारा final answer क्या है? Option 1, that is restriction endonuclease led to the discovery of genetic engineering. Hope you understood this well. Best of luck. Thank you.